So, let us continue the active and passive immunity and vaccination and different aspects. So, in my last lecture you remember, like we are talking about the same topic. Today also, I mean in this lecture also we are going to continue. Hope we will complete the up to passive immunization and the summary part in this lecture. Let us see. So, this is the acknowledgement for use of PowerPoint presentation from Genoa's book. So, now we are after this attenuated vaccine, now I am going to talk about the conjugate vaccine. Okay. Conjugate vaccine have been developed as a result of linked recognition between T and B cells. I just mentioned a little bit in the first lecture on this topic, passive and active vaccination process. Like if we would like to activate the immune system against the polysaccharide, what is going to happen? Because polysaccharide T cell is not going to be activated by polysaccharide. So, what to do? And so, new conjugate vaccine idea came, and it was a very nice and clever idea that was proposed and used. And what are the bacteria? This Neisseria meningitis, Streptococcus pneumoniae and Haemophilus influenza. Okay. So, these organism or these bacteria actually produce some polysaccharide against which we need the vaccination. So, how it happened? How it is done? So, you see we are coming back to immunology, how immune system react. So, this is B cell, and say for example, any particular B cell receptor which can recognize this polysaccharide which is outside the bacteria. Okay. So, B cell receptor can recognize, but if it can bind, but these binding, what will happen after this binding, it will internalize that part and then it will be degraded. But in this degradation, carbohydrate is not going to fit here. Which part will fit here? This red part. Red part is the conjugated part. So, what is that? This is titanous toxoid. Okay. So, titanous toxoid has nothing to do with this in this particular case, but we can have double protection. So, titanous toxoid fused with that particular carbohydrate are taken up by the B cell, processed and presented by MHC2. Okay. Presented by MHC2. You see the MHC2 is presenting the red part actually. This red protein part is presented by MHC2. So, this MHC2 presentation will attract helper T cell, it will bind, activate, and I am not going to be detailed on how the T cell will activate B cell, you already know that. Okay. It will give signal. So, this particular B cell which has a receptor against carbohydrate will be activated by presenting a protein, and that is why it is called conjugated protein. That means, carbohydrate and protein is conjugated. So, T cell is seeing the protein part and activating the B cell is producing antibody. Where this antibody is binding? Antibody is binding to the bacterial polysaccharide. Okay. So, that is how this particular conjugated vaccine developed against these three bacteria what I just showed, meningitis, Streptococcus and Haemophilus influenza. Okay. And this is another, the conjugate vaccine development, this is particularly this immunization against meningococcus, okay, the meningitis. So, what happened? There are two serogroups, this is a story of England and Wales. So, two serogroup of B and C is there. So, both used to create problem, cause disease. Okay. So, when the conjugate, this is just to see show the effect how good is this conjugate vaccine is. So, this is the number of cases in different years from 97 to 2004, it is going up and down because it is a natural infection going up and down. And in 1999 C immunization against C conjugate vaccine, serotype C conjugate vaccine introduced. You see, if you consider these two groups, serotype group B and C, B remain intact. So, after 99 there is no change in serotype B, it is as usual, like some year it is going down, some year is going up. Okay. But what happened to serogroup C? After introduction of the conjugate vaccine, the number is gradually grow down. In 2004 it is very minimum, I am sure now also it is, I do not have the data, but it is very minimum or no infection is there, or even if it is there, the number is pretty low. Okay. So, that means the seroconjugate vaccine against this meningitis is working. It is the real case. I mean, it is not just the laboratory or the animal model. So far we said what? We said whole cell vaccine, attenuated or heat killed, then subunit vaccine, so much and so on. But besides that, there is one more type of vaccine is also possible. It is called peptide based vaccine. Peptide based vaccine means let me draw that. So, suppose this is the protein, okay, and in that protein we have different epitope. Okay. We have different epitope. So, 3 epitopes are there. So, if I inject the whole protein, what will happen? Antibody will be produced against epitope 1, say epitope 1, epitope 2 and epitope 3. So, now instead of that, instead of purification, because many times what happened, purification or the production of the complete protein is really expensive and cumbersome. Okay. Pure protein, isolation of pure protein, in many cases it may not be possible for variety of reasons. Okay. This is not time, or I do not have the time to explain all these things. Variety of reason, complete or pure protein isolation, purification in large quantity is almost impossible for variety of reasons. So, that time what I can do? What I can think is, instead of the whole protein, I can just cut this region. So, it is a small peptide, okay, which has one epitope. You can have another. Okay. How do you know which part is the epitope? Many wet lab research you can determine which exactly the epitope sequence is. Besides that, there are many algorithm based softwares are available, where if you put the protein sequence or the primary sequence of the protein, it can predict you how many B cell epitopes are there, how many T cell epitopes are there. Definitely there are certain points how they determine it, but so few points are very simple and straightforward. Say one point I can tell easily, all of you can understand. Epitope, for B cell epitope, it must be hydrophilic, right, because it should be on the surface of the protein. So, this is one. The length, okay, number of amino acids, okay. There are many others. I mean, I am not going detail here. Anyway, so what happened? So, this epitope prediction you can do by bioinformatic analysis or immunoinformatics. You can test it in the lab and figure out that this epitope, I mean this peptide, if I take, or this big peptide if I take, that can be a good antigen. But this peptide cannot elicit immune response by themselves. Okay. So, you need to add what? For any immune response, you need one B cell epitope. Along with that, what you need? You need a T cell epitope, because until unless you have a T cell epitope, it cannot act as immunogen. So, many times what we do is we add another big protein called carrier protein. Okay, this is a peptide. This is the peptide, we add a carrier protein. Along with that, and with adjuvant, which is very important in that case. So, carrier protein, peptide combination and adjuvant together we have to mix. So, peptide alone cannot elicit the immune response. So, this is slightly disadvantage here. And adjuvant, we already told, in case of human, only approved adjuvant is alum. Okay. But in case of animal, because we need to raise antibody for many reasons in animal, for regular day to day research activity, for passive immune treatment, that will come later. So, there are another very important and very common adjuvant is there, it is called French complete adjuvant, which contains oil water emulsion, oil in water emulsion to make the micelle, and another thing is used, the heat killed mycobacterium. Actually, whole mycobacterium is not necessary, peptidoglycan muramyl dipeptide is important, okay, and the glycolipid tetrahalose mycolate is important, but instead of that, instead of purifying them, the heat killed mycobacteria is mixed with oil in water emulsion, and that is called Friend's complete adjuvant. What we do actually for animal treatment, when we inject or administer the antigen first time, we do complete, okay, the whole thing here, okay, the whole thing here we inject. So, we mix this mixture along with the antigen plus Friend's complement adjuvant we inject. But for booster dose, we just use oil in water. So, first injection complete, and second injection or the booster onward we use incomplete adjuvant. What is incomplete? We do not give this mycobacterium part, only oil in water. So, this is only in case of animal, not in human. In human, only adjuvant is alum. Okay. That I already discussed. Protective immunity can be induced by DNA based vaccination, like the you can inject directly the DNA. Okay. Vaccination and checkpoint blockade may be useful in controlling existing chronic infection. That is important. What people are now doing it, now people are doing dendritic cell vaccination. Okay. So, dendritic cell loaded with the antigen, okay, doing it outside and injecting it, which actually what they are doing it, they are inducing the different cytokine expression. So, activated dendritic cell cytokine activation, which normally the immunocompromised or infected person, this is a particular case of human HIV infected individual. So, what happened, many cases it happened if there is a weak response, then this, but if the response is good, you see the viral load is much less. Okay. So, that means the dendritic cell mediated vaccination is also started, and when it is shown that the vaccine, what is happening in that case, that dendritic cell mediated vaccination, in fact, this is the weak response, the IL-2 production is much less, where the response is good, IL-2 is more, interferon gamma is more, okay, in strong response. So, that means these particular dendritic cells vaccines, this is all very new development of the vaccination. It is the latest trend of vaccination. It is just not, because classical immunization process, purification of the protein and injection is not enough for most of the disease. That is why people are trying several other strategy. Okay. So, this is the reason. And a recent trend is the RNA vaccination. RNA vaccination is a lot of lab all over the world at this moment are trying to protect COVID infection or COVID-19 infection. Okay. At least next few years, I know this is the COVID 19 infection year or the pandemic year, everybody we know, but even for next several years we will remember what is COVID. So, what is RNA? So, not even the DNA to RNA step. So, directly for RNA virus, taking the positive strand RNA and making a nanoparticle as a carrier, injecting the RNA directly, so that next step protein and the immune system can be active. So, people are trying the RNA vaccination also. Okay. So, these are few things what about vaccination and vaccine production. So, before going to the passive immunity, let me talk about vaccine production. Vaccine production is what? Vaccine production is production of antigen. Okay. So, if it is whole cell, you have to grow the cell. If it is whole attenuated, what I told you. So, you have to do all this thing. If it is heat killed, you have to grow the cell and then either heat killed or inactivate by the chemicals. If it is a recombinant protein, you have to produce a recombinant protein. But the problem here, though I told a little bit in between the discussion, the problem it is the amount, the total number of vials we need to cater the whole world. Okay. In case of COVID-19, but in some disease what happened, it is not happen every part of the country. Some part it is very much acute problem. In some country you do not see all these disease and not all the population are affected by that. Okay. But say polio, all the, I mean, to eradicate polio from the whole world, you have to vaccinate all the children, and you need lot of vaccine for that. So, that production you need a big industry, you need a big fermenter for that to grow the cells, and you need a huge system to purify the protein. Okay. From that grown, suppose you are producing a recombinant protein in bacteria. So, you have to grow thousands of liters of bacteria, and it is not like lab, okay, we do just centrifuge 5 ml or 10 ml bacteria in a centrifuge small tank. Just imagine 10,000 liter you are growing, not much, 10,000 liter is a small scale, 10,000 liter medium you grown bacteria, you are centrifuging it. So, everything is huge. It is just not scale up is very easily. Okay. So, upstream and downstream is too different. Purification is very tough, precipitation of the cell is very tough. When you say 200 ml 2 liter culture is fine, but 2000 liter, 20,000 liter, 200,000 liter is not. That problem is there. Maintenance is a problem, supply is a problem, and administration is a problem. So, we need lot of trained manpower to do that. So, it is just not the discovery of the vaccine, we have to improve the whole system, so that the vaccination is more effective and will be successful to remove or eradicate the disease in near future for almost all infectious disease. I am definitely hope, and some of you at least will study this in future, do research and do something for vaccination. It is doing something for whole community, whole world. Okay. So, next what I am going to talk about, this was whatever I told is the active immunity. Now, the passive immunity. Passive immunity is what I already told. Passive immunity can be natural versus artificial. Passive immunity means when antibody is raised somewhere and you are getting a ready made antibody or T cell to protect the disease directly. Natural means it is very common, all of us know the passive immunity what happened, when happened. When the baby is in the mother's womb, what happened? Immune system is developing, right. So, mother's antibody are protecting the baby inside the mother's womb. Not only that, I mean even the immediately after birth the immune system is completely ready, but they are not immunized and can protect immediately. So, immediately after birth, before the breast milk started, there is another very thick material mother's body produces called colostrum, and this one contains lot of antibody, at least for few days it can protect the baby. So, here what is happening, this antibody produced in mother's body protecting the baby. This is also a passive immunity. Okay. And the breast milk also contains some, and gradually going down and slowly the amount of antibody reducing in the breast milk also. So, most probably for 6 months a little amount of passive immune protection is coming from the mother. That is natural immunity. Okay. And what is artificial passive immunity? Artificial passive immunity means say snake bite. What we do? We use anti-venom antibody, which is already raised in other animal. Other animals means mostly the horse. Why you use horse? Because horse is a big animal. We can isolate more blood, more antibody. So, normally what happen, I think I told in one of the class, if I remember, that suppose tetanus or rabies. Okay, what happened? So, somebody got bite by a dog, immediately we go to doctor for vaccination, but if that dog is severely infected with the rabies virus, so that bite transmit lot of virus. So, if you give immediately the vaccination, what will happen? Vaccination will not work immediately, it will take time, but by that time the disease may progress. So, what we need? We need vaccination, so that if anything happen, the vaccination will protect. But if something happen immediately, vaccination cannot protect. That is the difference between active and passive immunity. So, we need some antibody which is already made somewhere to inject into that person who got the bite to protect if there is any virus immediately transmitted to the body. So, that case both vaccination like the active and passive immune protection is required. So, vaccination of the rabies which is discovered long back is very effective, will be injected, as well as some antibody against the rabies virus which will not cause the disease will also be injected. In case of snake venom, there is no question of active immunity. We need immediate neutralization of the venom or toxin. So, we need some antibody to be injected and that antibody should be ready, right. That is raised in horse, and this is the artificial one. Okay. So, this is the artificial one. Neutral one we discussed and artificial one also we discussed. The first success story of the artificial passive immunity is the diphtheria toxin. Diphtheria is very dangerous disease for the kid particularly, even for elder age, but normally it happens to kid, that obstructs the throat and the airway. So, the breathing problem and ultimately death. Okay. So, what happened, I mean people tried, I mean the first experiment is in 1890, long back. Okay. S. Kitasato and Emily von Behring, what they did, they immunized the guinea pig against diphtheria with heat treated blood product from animals that had recovered from the disease. Okay. So, one animal which got diphtheria, that blood was heat inactivated and injected into another animal, and what they saw? They saw the protection. Okay. They saw the protection, and in fact, that was the discovery of antibody. Okay. So, let me go back. That was the discovery of antibody. So, what happened? They figured out that the blood produced something which can neutralize or which can bind with diphtheria. They named as antitoxin. Okay. So, that time antibody was not discovered. Okay. They named as, both of them received Nobel at I think 1903 or some year like that. And if you find the Nobel laureate history, you will find many physiology and medicine Nobel are from immunology background. Okay. With respect to all other branches of biology, if you see the number of Nobel laureates in immunology is, I never counted that way, but it may be the most of them are immunologists. Some will be biochemists, some will be protein chemists, that way immunologists are really in good number. That discovery is most of the discovery. So, what happened? Not only they restricted this into animal, that time the rules, regulation and the act was not that strong. So, this antitoxin, they figure it out, they can protect the diphtheria infected individual also. Okay. So, that first discovered that secondary or passive immune protection is possible, and this is how it is this passive immunity discovered, and after that all snake venom, all passive immunity and then rabies, whatever I just told, this one is discovered. But what is the advantage of and disadvantage of passive immunization? Advantage definitely is there. Just if you consider the rabies virus infection or the dog bite, it gives immediate protection. Okay. Somebody got a cut, go to hospital, get tetanus vaccine, that means if something happened. But if some patient come with tetanus infection and with the symptom, that means toxin is spreaded all over the body, you do not have the time for vaccination. So, you need immediate the administration of the anti-tetanus serum or the antibody, so that all tetanus toxin neutralize. That only can save you. So, that way active passive immunization is much faster and give you more quick result. So, that immunization cannot do that. Okay. But that there is a disadvantage. What is the disadvantage? Normally we develop the antibody against all this toxin and other virus in other animal, and these animal antibody may act immediately and neutralize and do its job, the person may be survive, but our immune system will find that horse antibody as a foreign protein. So, that I discussed once before also during idiotype and anti-idiotype. So, that antibody will be treated as foreign, our body will produce antibody against the horse antibody. So, gradually it will be neutralized. By that time purpose is over, but what will happen? That will not last for long time, that will not last for long time. Okay. So, passive immunization that way is faster, but we cannot avoid that. Okay. There are many cases, and here is also the similar problem, like you have to have lot of antigen to serve, like just for example, there are so many dog bite in country like India, there is so many snake bite during the rainy season. Okay. So, just to cater those, and they are not going to do their job, or that they are not working well throughout their life. So, every passive immunization, the antibody also has a half life or the life, or rather expiry date. So, after the expiry date, so normally what happened, so if it is written, what we do see in the medicine, the expiry date, if it is written in any vaccine or this kind thing, like through, this is also just for through August 2020. So, if it is written on this vial, that means September 1 we are not supposed to use, whether it is good or bad we should not care. But if it is saying it is like fixed date 16/8/2020, if the expiry date is written like this, okay, fixed date, that means on 17th August we are not supposed to use. So, this is how specific it is. So, so when you are producing this, any company, those who are producing this, first they have to produce a lot. Second, if there is not used in time, we have to throw it. Okay. So, there are a lot of loss in case of this kind of passive immunization material. Vaccine, there is a demand. Okay. And passive immunity, the antibody produced in different company, definitely there is a demand, not only snake bite, okay, scorpion bite is also not easily handleable. So, we have a antibody to, there are a lot of scorpion which is very toxic, okay, mostly are toxic, but some are very toxic, death, death is the end result. So, in that case we need this kind of thing. Okay. So, what happened? The passive immunization is not stopped here. Now, people are trying how to avoid this animal, to make the antibody in the laboratory, so that it will be more humanized. Humanized means human will not figure it out as a foreign and develop antibody, so that it can last long. Okay. And that is actually the future trend. Okay. So, this is the future trend, and this future trend we are going to discuss in the next class. And what will come? Many of you may know the monoclonal antibody, antibody engineering, there are many things are there. So, in next lectures we are going to discuss how we can generate antibody in the lab, purify it. So, we do not have to use the animal or harm the animal, and that is not only the single point, that not the harm animal, as well as that antibody will be humanized. So, that human will not figure it out, or human immune system will not figure it out them as foreign. Okay. Bye. So, see you in the next class.